Okay, this is Steve Rosenberg, and I want to thank the organizers for giving me the opportunity to give this talk. I'm going to talk today about the development of two gene expression tests for heart rejection and coronary artery disease that I've been involved with. And we'll take you really through, it's a tale of two product development cycles, and that really covers gene discovery, development, and then clinical validation and utility of these tests in clinical practice. So just conflicts that I have. I'm currently an employee of CardioDX, and I previously was an employee of a company called XDX. So let's talk a little bit about peripheral blood gene expression and molecular diagnostics to sort of level set everyone, so that those of you who aren't familiar with this can get a good sense of it. So if one's thinking about the differences between the utility of gene expression and genetics in clinical diagnostics, there needs to be, remember, of course, that genetics is generally static, at least to a first approximation, and really reflects heritable lifetime risk. It's very easy to measure today. And at least most of the studies and in most clinical situations, common genetic variation only reflects a modest proportion of overall disease risk. If one looks at gene expression, on the other hand, it integrates both genetics and environmental effects, such as diet, drugs, other risk factors. It's a little bit more difficult to measure. It's more subject to sampling and methodological challenges. It has an advantage because it's also time dependent. It monitors what's going on today with you, and hence it has the possibility of monitoring disease progression. One of those issues is that it is a continuous measure. You have a wide variety of gene expression values possible, rather than discrete values in your genetics. And we'll talk about two examples of blood-based multi-gene expression tests today. So if we look specifically at the peripheral blood gene expression as a window on the immune system, one can imagine that in the extracellular fluid in blood, plasma or serum, there are a lot of potential biomarkers, including DNA, microRNAs, protein markers. And these can be derived either from circulating cells or other tissues, such as the endothelial cell wall, vascular smooth muscle cells, or other organs, even including the microbiome. What we'll be focusing on today are peripheral blood cells, which really represent a very complex mixture of cell lineages and specific cell types, starting from hematopoietic precursors all the way to terminally differentiated cells like platelets or red blood cells. And with respect to gene expression, each of these cell types and lineage subpopulations have different patterns of gene expression. The patterns are dynamic and they respond to both the ingrained genetics as well as environmental factors, including pathological conditions. And with respect to pathological conditions, one can see variations in both proportions of cells, distributions of lineages from precursors all the way to terminally differentiated, as well as specific gene expression patterns within a particular kind of cell type. And lastly, an important point to recognize is that if you have an immune disorder, changes in peripheral blood gene expression may actually be a direct reflection of an immune response, or they can be an indirect reflection of the response of those circulating cells to a cellular or tissue insult. So now let's talk about the first example I'll cover today. That is the development of a blood-based gene expression score for heart transplant rejection. So the first heart transplant was done about 50 years ago now. And limitations in organ supply still have prevented immunological matching for donors and recipients. For example, there are only about 2,000 transplants done per year in the US. Currently, there's 3-drug immunosuppressive therapy for these patients once they've been transplanted. And the 5-year survival is very good. It's over 70% if the patients are rejection-free. But longer-term survival is often compromised because they are on this long-term immunosuppression. leading to infections and, in longer terms, lymphomas and leukemias. And the way you monitor rejection in these patients is by what's called an endomyocardial biopsy, which is an invasive procedure that's done up to 12 times in the first year post-transplant. It's an invasive procedure. There are sampling issues because it ends up only sampling a specific location in the myocardium. And depending on which pathologist and at which time interprets the image, you can get different results. A multi-center study was started now a dozen years ago or so at 8 major transplant centers. And what was obtained in this study were blood samples, in fact, mononuclear cells, a subset of all of the cells in the blood, as well as the results from an endomyocardial biopsy, clinical data on medications and outcomes, and a total of 630 patients, roughly speaking, were enrolled, and about 5,000 samples obtained. This was by far the largest study looking at genomic information in transplant that had been done. Some of the challenges with using endomyocardial biopsy as a gold standard in the development of the test are listed on this slide. So the transplant pathologist scale that was originally developed is really based on the appearance of infiltrates in the myocardium and not really based on a linear or monotonic increase in disease severity. What is agreed is that grade 3A rejection, now called 2R, is really the threshold for treatment with bolus IV steroids to reduce the likelihood of transplant rejection with hemodynamic compromise where the heart muscle no longer pumps effectively. In the cargo study, 3 very experienced transplant pathologists examined over 800 samples. And in a paper they published, which I'll refer to later, they showed there was a very limited concordance between them. This then illustrates a common problem with an imperfect gold standard, which has both statistical and clinical imports. So in this study, the subsequent work to defining what a rejection was limited to at least 2 pathologists out of 4 total reads, identifying a sample or patient sample as grade 3A or greater rejection. And this, as I mentioned, is a real challenge for developing a test with this as a gold standard. So the overall scheme for the discovery and development is complex and is shown on the next slide diagrammatically. Really break it up into 3 parts. Initially, there were microarray studies that were done. This was actually a custom microarray before the wide availability of large-scale whole transcriptome arrays. And also, because of the limitations of those arrays, literature and bioinformatics were also used. About 300 patient samples were subjected to these microarrays. And from that, about 100 genes and about 150 from the literature were pulled together as a gene discovery group where they had shown statistical significance in separating people with 3A rejection from those who did not. These array-based genes were then converted to PCR assays, and those were then looked at in an additional 150 samples, yielding about 68 candidate genes. These genes were then put together using a statistical method to construct what are known as metagenes, where the correlation structure of the gene expression data was used to reduce the dimensionality. This then yielded about 20 metagenes that were used along with the individual genes in algorithm development. And this finally led to an 11-gene classifier. You could estimate the performance of that classifier on the patient samples that was used to develop it, but the much more rigorous way and way that's really required is to use an independent validation set, and that's what was done subsequently. The next slide really covers what I just said. Specifically with respect to the algorithm development piece, these 68 genes and 20 metagenes were used in algorithm development. A number of different statistical methods were used, including logistic regression, random forests, and linear discriminant analysis. The final algorithm was developed with LDA, as it's known, It's the most rigorous method for estimating performance by cross-validation. And we use this method then to both select terms and weights, and also to add additional terms and see whether adding additional genes or terms would improve the performance. The actual biological basis of the algorithms is shown on this slide, where you see terms which are involved in steroid responsiveness. Steroids are used in the treatment of the disease. Platelets, hematopoiesis, probably reflecting the fact that the lineage has changed in this setting when you have transplant rejection and you have bone marrow stress. And then there are a number of genes involved in the activation of T cells, which are really the marker and the hallmark of transplant rejection. Though there was not required that these genes have known functions which are important in transplant, it obviously is a useful thing to get support for such a test downstream. So for validation, there was first a primary validation set where the patients were enriched for rejection samples, 63 total patients that were completely naive to any of the development and discovery work, and 31 rejections and 32 quiescent patients, and then a secondary validation in an additional larger set of independent samples. And this was then coupled with a pre-specified analysis plan and a score threshold. When you look on the next slide where we have the primary endpoint, this is by ROC analysis, where you plot the sensitivity or true positives versus 1 minus the specificity of false positive rates, and one measures the area under the curve, where a test which is no better than random falls along the diagonal or has an area of 0.5. And in this case, the area under the curve, or AUC, was 0.72, much better than random, sensitivity of 84%, quite good, rather modest specificity. And it turned out that the time post-transplant, when steroids are being weaned, is a large confounding variable in this study. And when one looks at performance later post-transplant, in fact, performance of the test is considerably better and higher thresholds can be used. So that's illustrated here, where we see the original validation data with all patients post-transplant on the left with an AUC of 0.72, and if you look at either 6 months or 12 months post-transplant, you see the AUC goes up significantly to 0.8 or 0.86. All of these results, however, are challenged because of the uncertainty in the true rejection status of the patients due to the complexities of endomyocardial biopsy. So in order to look at a less ambiguous endpoint, a second study called the IMAGE study was started. In this study, about 600 patients from 6 months to 5 years post-transplant were enrolled in 13 centers, and they were randomized to either gene expression testing as their major mode of care or endomyocardial biopsy. So it was a head-to-head study comparing these two. The primary endpoint was non-inferiority for the gene expression test in terms of patient outcomes, where here we're looking at death, retransplantation, or rejection with hemodynamic compromise as the primary endpoints. And the secondary endpoint where it was examined whether there was a difference in the number of biopsies performed. So here we have a much less ambiguous endpoint and one which is clinically, obviously, clearly relevant. So the next slide shows the patient flow. And it turned out this was a relatively difficult study to enroll, starting with about 3,000 patients. About 1,200 were invited to participate. Finally, about 600 of those were excluded. So we ended up with 602 patients enrolled. So that raises some questions about whether this population is overall representative of the entire transplant population or not. And then you'll see at the end it looks like it probably is, but it's not so clear. These patients were then randomized, 297 in each group, and there was quite good follow-up with 2-year follow-up, looking at the endpoints I mentioned earlier. So what happened? So if we look at the next slide, what we can show and see is that in the upper panel, where it's looking at events as a function of time, what one sees is that overall the event rates, which were around 15% over 2 years, were no different between endomyocardial biopsy-based care and gene expression. based care. And in fact, survival curve, which is shown on the bottom, also shows no significant difference. What I don't show here, but was a secondary endpoint, was that the number of biopsies was reduced by 2/3 in the gene expression-based arm. So they're a very nice study. And really, this is the only study which has been done where a randomized, controlled, and blinded study was performed comparing gene expression-based testing in any really clinical indication with standard of care looking at long-term outcomes. And the key references for this work are shown here. The first one being the pathology work I mentioned at the beginning. The second is the discovery, development, and validation of the gene expression test. And the last is the image study results published in the New England Journal now 3 years ago. So we're going to switch gears now from heart transplant rejection to a gene expression test for obstructive coronary artery disease. As I mentioned earlier, there are only a couple of thousand transplant patients seen each year for heart transplants. There are millions of people who have symptoms suggestive of obstructive coronary artery disease who are seen by physicians each year. And that's why this test was developed. So if we look on the next slide, why did we think that such a test was possible? Well, it's known based on work originally published by Russell Ross and others that inflammation is a key driver in the development of atherosclerosis. Other workers have shown that if you look in the diseased vessel wall, you see changes in gene expression in people who have atherosclerosis or coronary artery disease. Multiple groups have shown that the example I just went through is a good example of changes in peripheral blood gene expression that correlate with inflammatory and immune-mediated disorders. And hence, we ask the question, can gene expression in circulating cells reflect the presence of coronary artery disease in the vessel wall? Well, why would you want to do this? Aren't there other ways to do this? Well, it turns out in a landmark paper which was published in 2010 in the New England Journal, where the National Cath Lab Data Registry was interrogated about 400,000 patients who had been sent for elective coronary angiography. Turns out only little less than 40% of them, 38% in this study, had in fact been shown to have obstructive disease and in fact had any need to be in the cath lab at all. So more than half of these patients did not need to have an invasive procedure. And this was despite the fact that over 80% of them had received non-invasive imaging tests or diagnostic tests prior to the referral to catheterization. So clear and unmet need, we can do better than this. So in the development of this coronary artery disease gene expression score, multiple independent cohorts were used in the discovery, first phase, development, second phase, and algorithm validation, third phase, where in the initial part of the study, and single center cohorts were used that were retrospective, and then prospective multi-center studies were used subsequently to end up with the development and validation of the test. Initial gene discovery was done on a small set of microarrays. In this case, these are not custom arrays, but ones from Agilent. About 50 candidate genes were identified, and an additional 56 were taken from the comb from the literature. RT-PCR assays were then developed. And these were then tested in subsequent independent cohorts, one from the same German cohort and another from a set of samples from a registry at Duke called CAD GES. And what was found based on these results are shown on the next slide, where what we've plotted are the significance of the gene based on PCR assays in the Duke CAD GES cohort versus that in the German cohort. And what you can see is there's a group of genes which are found in the lower left, which are not significant in either one. And a total of 27 out of the 106, in fact, were significant in both cohorts. If everything was perfect, everybody'd be on the diagonal. But in fact, in the Duke cohort, this was using whole blood. And in the Siegberg cohort, they were using just CPT tubes. So there were additional genes present in the Duke cohort, which are more significant. which are those significant in Seaborg or even more significant there. So that was one of the observations made. Second observation made in this study was in the CAD-GES microarrays, was that patients who had diabetes, in fact, had different genes dysregulated if they had coronary artery disease compared to those without. Just like in the previous slide, if the same genes were dysregulated in both cohorts, you would see genes along the diagonal. What we see here is no genes of significance between both diabetics and non-diabetics. And independent groups of genes, in fact, for diabetics, they're largely from natural killer cells, and they're largely driven by neutrophil genes in the non-diabetics. So this led us then to limit subsequent test development to non-diabetic patients. We then enrolled a multi-center study called PREDICT. Eric Topol Scripps was the PI. And this was a cath lab population where patients who had already been clinically referred for an invasive angiogram after clinic visits and possible non-invasive testing were screened, enrolled, and a large amount of clinical data was obtained. Research blood samples were obtained, and then they went on and had their invasive angiogram. So what we had in this study then was paired results of an invasive angiogram, a picture of these patients' coronary arteries, as well as blood samples. The angiograms were then read in a core laboratory by a computerized method called quantitative coronary angiography to reduce variation, and RNA was extracted from the blood samples. So in the gene discovery phase, microarrays were completed on about 400 patients. We found the largest effects overall in gene expression to be due to the patient's age. The cell type proportions, we had complete cell counts in all these patients, as well as diabetic status. So we limited this to non-diabetics. We derived sex-specific age dependencies for coronary disease risk. And we picked 113 genes from the microarrays to be measured by PCR in an independent cohort. We used a similar approach that we talked about with respect to the heart transplant rejection, where we used metagenes. In this case, we used ratios of metagenes to give general and cell type specific normalization. And the method of choice for algorithm development here was logistic regression, where we used ridge regression finally to get the final weights. And this was described in a paper that was published a couple of years ago. In terms of the microarray work, the initial studies from the Seaborg and CAD-GES discovery cohorts were heterogeneous in terms of the methods of RNA extraction used, the kind of tubes where the blood was obtained in one case, CPT tubes, in another case, whole blood packaging tubes. The case control definitions were subtly different, as were the inclusion criteria. So this was a possible source of variation and possible confounding variables. We then went on to extend the discovery work to the PREDICT study, where we had in the meantime developed an automated and very reproducible robotic method for RNA extraction from whole blood. We used quantitative coronary angiography to define case control status, and we used a paired design where the sets of cases and controls that were matched with respect to age and sex were in fact run on the same microarray chips. And finally, this was restricted only to non-diabetics. And as you can see on this slide, whereas we had a modest number of genes in the original two single center cohorts, 500 roughly and 2400, in this last multi-center derived cohort where we had made these process improvements, same size cohort, but we were getting 2.5 to 3 times as many genes as significant, almost 6,000. So we used all of these data with a bias towards that last PREDICT set to derive these 113 genes, which were then developed PCR assays for. And these were then run on an independent set of 640 patients from the PREDICT study to use in the development of the final algorithm. One of the things that we found in this data set was that, as we had suspected, there were quite significant differences between men and women. What we show here is the percentage of patients, women in red, men in blue, who had either no significant coronary disease, clean coronaries, on the left. And what you can see is almost 40% of the women had no detectable disease, and about 20% of the men. And conversely, when one looked at obstructive coronary disease, meaning a 50% blockage or stenosis in any major coronary artery, only 1 in 5 women had such a blockage, whereas it was almost half with men. So this clearly said that you needed different kinds of effects to measure in the two genders. So I've gone through this already once, but we use that data then to derive sex-specific age dependencies for coronary disease risk. We used the microarray data that was then run on the 113 genes as RT-PCR to develop this large matrix of 113 genes by 640 patients to then use as the algorithm development set, using a number of different statistical methods, including clustering as well as a variety of correlation data, and using some biological input as well. We derived metagenes. Metagene ratios were used to then come up with potential terms, and then logistic regression was used in a forward stepwise manner to pick the terms that were significant on top of the age and sex-dependent coronary disease risk factors. So what we ended up with then was an algorithm with 23 genes in it, and it contains 3 kinds of terms. The first is the gender-specific age-dependent coronary disease risk functions. Second are, because each cell type within all the lineages in blood has a distinct gene expression pattern. In fact, you can separate the gene expression pattern in cells by cell types pretty readily. We use that to, in fact, be able to essentially count specific kinds of cells, like natural killer cells, for example, in the blood, and have terms that reflect ratios of cell types. And then finally, we have genes which are up or down regulated within a given cell type as a possible kind of term. So if we look at the underlying biology behind some of these terms, some of the genes in the algorithm are shown here. In the upper left, you see a group of genes, including caspase-5 and IL-18 receptor-associated protein that are involved in apoptosis, inflammatory cell signaling. On the lower left are a group of genes which are upregulated in response to oxidative damage, oxidized LDL being a driver of atherosclerosis, as well as response to cellular necrosis. We see genes that represent specific cell types, namely those for T cells and B cells. And finally, another set of genes on the lower right, which are specifically expressed in the innate immune response and specifically upregulated in natural killer cells. So once we had this algorithm finalized and locked, what did the validation studies look like? And in fact, we ran 2 sequential validation studies in 2 different populations. The first study was an extension of PREDICT, where we used an independent cohort of patients from the same overall study that we developed the algorithm in. This is a prospective multi-center and blinded study. All non-diabetics, 39 sites here in the US. And quantitative coronary angiography by Alexandra Lansky, who's currently at Yale, was run on all the patients. Patients were either had symptoms suggestive of obstructive coronary disease, or they were asymptomatic but had a number of risk factors leading them to have being referred for invasive coronary angiography. They were all non-diabetic, and they couldn't have had a prior heart attack or revascularization procedure because you wouldn't need a test then to tell whether they had coronary disease. When we looked at this overall population, and the validation study was published in 2010 in the Annals of Internal Medicine, we found that 37% of the patients in PREDICT had obstructive disease, essentially the same as the 38% that was found in the large NCDR study. And this really suggested that this relatively small population of about 500 patients that we used for validation really was typical as compared to this much larger group. So we then set out a prospective validation study plan. We had a locked algorithm, as I've mentioned, an independent cohort of patients about 525 in total. We had defined the case control definition as 50% stenosis or greater in a major coronary artery in at least one vessel. And that represented about 37% of the patients in this group. We locked all the clinical data and completed on-site monitoring according to good clinical practices, ran all the RT-PCR. We had predefined quality control metrics for the PCR and a predefined analysis plan. The primary endpoint was ROC analysis. Could you distinguish cases from controls, treating the disease as binary, which in fact it is not, as we'll talk about later. And the other endpoints focused then on seeing whether the gene expression data would add to clinical factors, which can be used to estimate risk of obstructive coronary disease. And all of this was done by an external statistical group led by Nick Shork at Scripps. So what happened? So the primary endpoint, shown here in ROC curve, is sensitivity versus 1 minus specificity. You see an area under the curve of 0.70, much better than random, which was 0.5, highly statistically significant, and importantly, was independently significant in both men and women. In this next slide, we ask whether the gene expression score would add to clinical factors. The clinical algorithm here is one that uses age, sex, and the kind of chest pain the patients have, which, at least in men, is a very good indicator of whether you have likely to have obstructive coronary disease. And what we can show here is by ROC analysis that the gene expression score did add to clinical factors in a statistically significant way. This is a highly rigorous method for seeing whether you're adding value to a pre-existing classifier. And if you then look at the percentage of patients who have obstructive CAD as a function of the gene expression score, which is reported on a 1 to 40 scale, what you see here in a logistic analysis is that there's a nice overall dynamic range from less than 10% all the way up to almost 80% for this group. And at a threshold of 20% likelihood, about a third of the patients fall below that threshold, where the sensitivity and negative predictive value of the chest in this patient population, who had already been referred for coronary angiography, is 37%. So a significant fraction of the patients, in fact, were found in that low-risk group. An additional factor, which turned out to be quite fascinating, and what you found was shown in this slide, where we plot on the vertical axis the test score, and on the x-axis we have the max percent stenosis, or another measure of overall disease severity or burden. And what you can see is that even among those patients who do not represent cases that have less than 50% stenosis, so the 3 bars shown there on the left, if you have no detectable coronary disease, you have the lowest average score. And that continues to increase even with non-obstructive disease, and continues to go up beyond that with single vessel disease or multi-vessel disease. So despite the fact that we're treating this clinically as a case control, 50% or greater, you should go on and potentially have an angiogram. In fact, the biology tells you this is, in fact, a continuous variable, and the more disease you have, the higher your score is. So what are the limitations of this particular study? Well, there really were 3. First, in PREDICT, we had a group of patients who had already been referred for coronary angiography. And the question was raised, well, what happens in a lower risk population, which is really where this test would be intended to be used? Second of all, if you want to use this test as a rule-out test for eliminating downstream procedures, invasive and otherwise, you really need to know whether it's safe to do so, and you need patient outcomes and events. And finally, there's really no comparison in this study to usual care, which is done with a non-invasive imaging method, primarily consisting of nuclear perfusion imaging or myocardial perfusion imaging. So we went on to a second validation study in the COMPASS trial, which I'll take you through now, to try and answer these questions. And we also had 1-year follow-up data on events and safety from the PREDICT study. So COMPASS, we looked at an independent validation of this test again in stable symptomatic patients only. And in this case, they've been referred for non-invasive imaging, specifically myocardial perfusion imaging, and really that represents an intended use population for such tests. Secondary endpoint was to evaluate the head-to-head comparison of NPR performance in the real-world setting versus the gene expression score. Another prospective multi-center blinded study, in this case 19 sites here in the US, about 430 patients, number of core laboratories we used, and an independent academic steering committee led by Greg Thomas, who's former president of the American Society of Nuclear Cardiology, led the study. As I mentioned, symptomatic patients, non-diabetics again. They couldn't have known obstructive disease or prior heart attacks or revascularizations. So here's the study design. Chest pain patients who were referred for myocardial perfusion imaging. They could have typical or atypical symptoms. A gene expression score blood sample was then obtained, and then their routine stress myocardial perfusion imaging study was run. If that study was positive and it was clinically indicated, they went on to invasive angiography. And what was a novel factor in this test was that if the myocardial perfusion imaging study was negative, and they normally would not have any further workup, a CT angiogram was obtained. The reason for doing this was that we would then have gold standard anatomical data on every patient in the study, independent of whether their MPI was positive or negative. We then used these anatomical data and took them to core laboratories to take out variation between individual readers. The invasive angiograms were then read by quantitative coronary angiography, as in PREDICT. The CT angiograms were read by two independent CTA core lab readers led by Szilárd Forrós. And in the case of MPI, we did the original interpretation based on the on-site reads, and then we also had core lab interpretation of those data just to make sure that we were not getting fooled by the MPI site read results. So, what did the patient flow look like? We had 537 patients enrolled. We needed to have a myocardial perfusion image. We needed to have a gene expression score. And we needed to have either a CT angiogram or an invasive angiogram for every patient in order to qualify. What you can see here is of the 537 patients, about 100 did not have the CTA. Those were patients with negative myocardial perfusion image who declined to have a CT angiogram. There were a few where the MPI images were not appropriate for reading, and only 3 who did not have a gene expression score. So 431 patients were evaluable that had both MPI, GES, and gold standard. Interestingly, almost 90% of them had negative MPIs, so this was quite a low-risk population. Only 48 had positive MPIs. If you look then on the upper part of the curve of the 383 with negative MPIs, almost all of them had CT angiograms, but a significant fraction then went on to cath. Matter of fact, a total of 46 of the cases who had 50% or greater stenosis had in fact negative myocardial perfusion imaging studies at study entry. For those who were positive for MPI, a little more than half of them went directly to cath, and about a third of them in fact had obstructive disease when one looked at the gold standard anatomical data. So a lot of patients with negative MPI scans. If you look overall at the study, and this summarizes the case control status in the negative and positive MPI groups, what you can see is that if you had a negative MPI, almost 90% of the patients did not have obstructive disease, and only 12% did, but that 12% represents a significant amount, about 2/3 of the cases, whereas since only 11% had positive scans, although the percentage is 35%, that represents a minority of cases. So MPI was had poor sensitivity in this study. But the diagnostic yield at cath was 35% with a positive MPI scan, which is similar to what was seen in the NCDR study I mentioned to you earlier. So if one looks at ROC analysis, then comparing myocardial perfusion imaging in this study with the gene expression score, you've seen this kind of graph before, what you can see is that here's the dashed line representing random, MPI site read was a little better than random, 0.59 AUC. Core lab went up a little bit to 0.63, but the gene expression score had an area under the curve of 0.79, a bit better than what was seen in the PREDICT study, where it was 0.70. And in fact, it outperformed MPI by ROC analysis in this study. If one then compares the gene expression score to a couple of different clinical factor algorithms, following along the lines of what was done in PREDICT, here we show that the gene expression score is in fact superior to either Diamond Forrester or another score, the Maurice score, in this population, which is lower risk. So one of the things that's important to remember in this analysis is that historically, when non-invasive imaging studies have been done, they've been focused on patient populations who have been referred to the cath lab to have an invasive angiogram, because that's your gold standard. And those who did not get referred clinically, in fact, were not looked at. So in a recent meta-analysis, looking at that referral biased population, the sensitivity of MPI was 81%. Recent estimates show that there's about an 8-fold to 10-fold referral bias of the likelihood of having invasive angiography if you have a positive versus a negative MPI scan. And if you correct this using some known statistical methods, the sensitivity for MPI drops from about 80% to 35%. Specificity goes up to about 94%. These results are very similar to what we saw in the COMPASS study. In COMPASS, referral bias is minimized because everyone with an MPI scan, whether it was positive or negative, was referred for further evaluation to get a gold standard view of their anatomy. And remember, in the core lab results in COMPASS, the sensitivity was 36% and specificity was 90%. So if we look overall at the validation study results, in comparing them in COMPASS, what you see comparing the gene expression score with site or core lab MPI, you see high sensitivity for the gene expression score, modest to low sensitivity for MPI, modest specificity for the gene expression score at a pre-specified threshold of 15, but high specificity for MPI. Negative predictive value, very good, 96%. This population is lower disease prevalence, so expect that to be higher than what was seen in PREDICT, and also superior to what was seen in MPI, which has a higher positive predictive value. But if you look at overall diagnostic accuracy as measured by the ROC analysis, the gene expression score was superior. So one way to look at all of this is to look at every patient in the study. This slide doesn't reproduce very well, but what we show here is core laboratory MPI results, either positive or negative, with jiggering to separate the points as a function of the gene expression score on the x-axis. And the points, each point represents an individual patient with either essentially no coronary disease, non-obstructive disease, or a case obstructive disease. What you can see is that the vast majority of patients with low scores, and a pre-specified threshold of 15 was used in this study, have very little disease. The 96% negative predictive value speaks to that. And as you go up in score, the amount of disease goes up. And you can see that, in fact, if you have both a high score over 28 and a positive nuclear perfusion scan, about 70% of those patients have obstructive disease, and almost all of them have some measurable disease. So this slide illustrates two things, that, in fact, myocardial perfusion imaging and the gene expression score are somewhat complementary. But if you're interested in the group of patients who don't have disease, the gene expression score does a much better idea of both identifying them and being highly sensitive to the presence of obstructive disease. So if we look then in sum at both the PREDICT and COMPASS validation studies, you can see that we got very concordant data. In COMPASS and PREDICT, the sensitivities are in the mid to high 80s, specificity more modest, about 50%. Negative predictive value was 96% in COMPASS, 83% in PREDICT, and that reflects the decreased prevalence in COMPASS versus PREDICT. The last thing to remember about this study was, well, what happens to the gene expression score relative to usual care, namely myocardial perfusion imaging, and in this case, the gene expression score outperformed that, both either site read or correlate read. So if we went and then look to safety and outcomes, what happens? So we've answered the first question, what happens in a population of lower risk? The gene expression score seems to perform well there. What about safety in terms of patients? So we followed patients for 1 year in PREDICT and 6 months in COMPASS. And if you look at this clinical diagram, which is shown on this slide, which is based on the COMPASS data, if you took the patients who had scores of less than 15, where the 96% negative predictive value was the case, that represented almost half the patients, 46% of the patients in the study. And you would say, well, those might not need any further testing. You would then take those who had elevated scores and have another test to further stratify them. If they had myocardial perfusion scan, most of those would be negative. They wouldn't have further testing. And a small fraction would go on to the cath lab. And in this case, you would enrich the population at the cath lab because none of these patients, in fact, who had low scores would go on to cath. And you would increase the yield of cath to almost 50%. In order to do something like this in the clinic, you need to know that these patients who are ruled out, who have low scores, in fact, don't have any adverse events going forward. So what we've illustrated on this slide is looking at low versus high gene expression scores as a function of whether the patients had either revascularization within a month or later revascularization or major adverse cardiovascular events, namely non-fatal myocardial infarctions, strokes, or all-cause mortality. What you can see is that in the column to the left, with the early revascularizations, less than only one, actually, of the patients out of the 199 in that group had a revascularization within a month, whereas for that group, about 230 patients who had elevated scores, the rate of revascularization was 20 times as high, 10.5%. And if one looked out 6 months at late revascularization and adverse events, there were none in the low score group. And all of the events, 3 in this case, were in the high score group. A similar pattern is shown in the PREDICT group with longer follow-up. There's a 3-fold increase in early revascularization for those patients who have higher scores versus low scores, and about a 2.5-fold increase in the event rate for those who have elevated score versus low scores over the following 12 months. If one looks at whether the gene expression score as a continuous variable was associated with increased event likelihood, it was in both studies in a statistically significant way. So to summarize these data, the coronary disease gene expression test developed through a series of microarray and RT-PCR studies. Test validation was accomplished in 2 multi-center studies. Primary role of such a test is to rule out a coronary disease etiology in patients so that they can then have their symptoms investigated in other possible causes. The high negative predictive value in the COMPASS study and consistent sensitivity in both studies enables patients to be ruled out with a high degree of safety and certainty. And that was reiterated because there were low scores, had little or no events, and can be followed up without subsequent referrals to cardiology or angiography. There are a series of papers published now on the coronary disease work, and those are summarized there. I want to acknowledge all my colleagues at both XDX and all the investigators from both the cargo and IMAGE studies, colleagues at CardioDX and the investigators in PREDICT and COMPASS, especially Bill Kraus, Solar Boris, and Eric Topol, as well as Alexander Lansky, John McPherson, and especially for COMPASS, Tim Bateman and Greg Thomas. And overall, then, to summarize, what I've tried to show you today is the development of 2 blood-based gene expression assays for heart transplant rejection and obstructive coronary disease that have been discovered, developed, and then validated in multi-center cohorts. There are significant limitations with the current standards of care as gold standards in test development, and in order to really get at clinical use, one really needs to then look at outcomes, which were done in both cases. The high throughput genomic technologies used to originally identify genes in these cases create now well-known statistical issues about multiple testing. These can be mitigated using sequential independent cohorts, where essentially one is getting rid of the noise and hopefully following the true signals, because the noise will randomize in additional cohorts and the true signals will continue to come through. You really need to show comparison to usual care to show that the genomic tests are favorable. And this can be done either in a prospective randomized manner, as was done in the image study, or in a single head-to-head comparison, as was done in COMPASS. I think with that, I'll stop, and I'm happy to take questions. So I'm sure I'm very clear in my talks, but not so clear that there aren't any questions. Well, as there don't appear to be any questions, perhaps we should finish this session. Don?
I'll go ahead and we'll stop it.
Okay.